genetics. And we welcome you both. I know last time we had planned to present, there were, a lot of things going on. So thank you so much for making the time to join us again and to be back with us. We welcome this evening Dr. Carter and Dr. Merrick, and they are the both co-directors of the Black Genome Project, as well as their respective associations with the public health departments at Yale University and the professor of genetics for Dr. Merrick at Washington University. And we welcome you. We have a lot to learn. We appreciate the topic that you're going to present this evening, providing us a bit of the history of genetics and research surrounding genetics and sort of where we go from here in terms of repairing in the future. So thank you so much for being here. I did just post a question answer so that it's visible on your screen, and you can submit those throughout the presentation. I'll compile them and we'll leave time for that at the end. If we don't have time to address your question, please be patient with us because I will still compile them and make sure we get back to you. Thank you. If there's any introduction you'd like to make before you start, or when you're ready, you may share your screen.
Awesome. Thank you so much. All righty, let's do this. All righty, that look okay to everyone?
Almost.
All
Yep,
right.
looks good, Chelsea.
Perfect. I guess I'm really just talking to you, Brett.
Yeah, that looks good. Cool. Well,
alrighty, awesome. Well, we are really excited to be here today. Many thanks to the CADASIL Consortium at UW-Madison for inviting us, and specifically to Elizabeth for hearing about our work at a different conference that we were attending and inviting us to share more about our work. Really excited to be here and just share a little bit about how Brett and I have been thinking about community genomics projects, but also how to really be thinking thoughtfully about repairing relationships, developing trust, particularly with communities that have had a history of harm. And so today our title is Prioritizing People, How to Develop Trust and Meaningful Relationships Through Community Engaged Research. We really just hope to kind of share with you insights that we found from our own Black Genome Project, but also really encourage, a lot of work that is being done right now in genomics is asking questions about diversity and asking about inclusion. And so I think just in this moment, it's really, I think, critical to just have these types of conversations so that we can share resources, share ideas, and figure out better ways to enhance our research. So I'm just going to get started just kind of sharing how Brett and I met, because I think we always kind of share this because we think it's important to the collaboration, because it's not every day that a anthropologist and a geneticist meet and decide to start a research project. So back in 2020, I was a graduate student at Washington University in St. Louis, finishing up my dissertation project, looking at ALS in Black and white communities, and really trying to understand more about misdiagnosis, access to care, clinical research trials, and how people were understanding their own illness experience. During that process, I had some really interesting stories with individuals that were in the clinic that were thinking about, why wasn't there a test for ALS? If you read this article that is titled "The Racial Thinking Behind ALS Diagnosis," I tell a story about a woman who came into clinic and was upset that she was kind of told by her physician that there was not a test for her. And particularly, there wasn't a test for her because she was a Black woman and helping to diagnose her ALS. And so I really kind of take to task in this article, thinking through the ways in which race becomes biology and how we talk about genetic ancestry, and how it also can kind of not necessarily, and how precision medicine isn't always kind of the equalizer that we hope it can be, but how it also can create further disparities and also leave people out. And so while I'm thinking about these questions, I'm also a scrappy grad student that's really looking for side jobs. And during this moment, in the COVID moment, there was a COVID seroprevalence project that was happening in the city of St. Louis, and Brett was one of the faculty organizers of the different sites where we were collecting blood from people in the community. And so I quite literally, he was my boss, and we didn't actually have a conversation in person until about 6 or 7 months after I actually was doing the job. And so once we finally connected and had conversations, we realized that we had a lot of synergies in not only our own backgrounds in community engaged work, but we were also asking these questions about what does genetics mean for the Black community in St. Louis? Are people interested and engaged in genetics and genomics projects already? And what does it mean for the future of precision health? And we realized that while a lot of pressure was being put on each of us in some ways to kind of start sampling and testing people for various genetic conditions, or simply just doing genome sequencing with the Black community, that the questions that I just shared kind of were still driving a lot of our inquiries around, do people want this? Is there interest? And how do we galvanize it? And so in a lot of ways, that is how the Black Genome Project was birthed. It was literally just two young researchers saying, hey, we've got some questions about some things and we're not seeing it in the literature, and we don't feel comfortable just going out into the community and starting to, start a research project that requires genomic sequencing. So where do we start first? And so for us, it really started kind of by identifying what the problems were, so we can think about how can we work in conversation with community to kind of circumvent these problems.
So yeah, Chelsea and I spent at least, I would say, 18 months. And we were fortunate given that we were both young investigators. Unfortunately, the pandemic had put pause on the work that we were supposed to be doing as young faculty, and it gave us some space to really think and plan our project. And so, in addition to a set of values and goals and objectives that we laid out, these problems were really at the forefront of putting this project together. And they really kind of revolve around the the advancements of precision health and precision medicine in the context of um, the history of genetics and genomics and science in general. And so the first problem that we identified, and others have written about as well, is that for precision health to really work, it requires an understanding of the full scope of genetic diversity that impacts health and disease. So, shortly after the initial human genome sequence was published in at the 2001, folks started going out and sampling genomes from across the globe, and a project called the Thousand Genomes Project, which really took this task on in the early 2010s, um, kind of showed for the first time something that we've always known given the history of human migration out of Africa, knowing that humans have existed on the continent for longer than they've been present anywhere else on the planet. And so therefore, there was an assumption that genetic diversity was actually greatest on the African continent, just due to genetic drift alone. And so these are some of the first genomics level data showing this. And so in the figure on the left, you're seeing samples from across the globe, and the dot plot on the lower left is showing the number of genetic variants from a reference genome that are seen in various populations. And those populations in blue, green, purple, and red are populations that are primarily of European descent and Eastern Asian descent, whereas those in orange and yellow are populations of African descent, including admixed Black populations in the United States. And what the consortium showed is that on average, genomes from people that are of African descent have more genetic variants than those from populations of European descent. And there's a couple things going on here. One is that the reference sequence that's being used is at the time ill-equipped to be aligning or comparing all of these diverse genomes to. But secondly, it is true that there's more genetic variation on the African continent, and these are evidence of that. So, in response to some of these data, we've seen things like the Human Pangenome Project, which has recognized the limitations of the reference sequence and has really expanded that reference to a set of genomes, and they're now doing rather than like single genome comparisons, they're comparing genomes to a graph of diverse genomes from across the planet. And then others in disease contexts have shown over the years and even recently that by incorporating cohorts of people that have wide genetic diversity, there's actually quite a bit of power in identifying meaningful associations between genetic variants and disease, and that this is an area that we should be continuing to invest in from a scientific perspective, because there's biology to be learned, and variants that have never been seen before that are informing us about new drug targets and new ways of diagnosing and treating disease. And so we've adopted this as a problem that we believe is key to the success of genomic medicine and precision health.
A second problem that we see that's, and all of these problems you'll see kind of all connect to one another and are related. But while we also understand that we need to and should continue to understand the full scope of genetic diversity, we also have to be mindful of the fact that if we want to diversify our genetic understandings of things, there has to be a reckoning with the fact that there are both historical and present day harms that have been done to vulnerable communities that we're seeing not showing up in our large GWAS studies. And so I think what this, the first graph on the left really reflects is that over time, since 2006, or even before that, 2001, like Brett mentioned, when we first sequenced the first genome, what we can see is that we have across the globe, we have a high representation of people from European ancestry. However, when we look at even some of the smaller groups in blue, green, and so forth, both East Asian, South Asian, but especially African, there's very, very low inclusion in that. And so that is something that we have to account for, but we also have to realize the why of why people are not being accounted. And so, the headlines on the right are just some of the many headlines, both in the past and the present, of the ways that, vulnerable communities have been impacted by forcible sterilization, not having access to healthcare because of structural and systemic barriers, or trying to take their own reproductive choices into their own hands, but not being able to because of how doctors have impacted, I shouldn't say doctors, how the biomedical system has gatekept and kept some people from getting access to reproductive rights and others from not being able to get reproductive rights. And so for us to think about precision health and really making strides across all populations, it's really important to think about the challenges, both historical and present day, that continue to create forms of exclusion. So despite that reckoning, however, we also have to really think about the fact that when we are having these conversations about representation in genetic diversity, that we have to make sure that we are not misconstruing this idea to also fall in line with very draconian racial science. And so, many of us have seen, I don't want to assume who's in the webinar today, but some of the research that has come out in the last couple of months, I would say, particularly the All of Us program, there have been, significant critiques about how the UMAP was used in their study, particularly because the ways in which it exaggerates and almost reflects an idea that ancestry is equal to biological race. And so while what this UMAP was really trying to show us on the left was the ways in which genetic ancestry impacted things, how it reads and how it can be interpreted, falls into tropes of what do Black Americans look like? What do white Americans look like? And also not only just kind of phenotypes, but of what they look like, but also how their biology must be uniquely different from a different racial group, which we know to not be correct. And what is really scary about that is the ways in which these types of beliefs that can be misconstrued without nuance, that do take genetic ancestry and map it onto social constructed views of race, is that we unfortunately see the ways in which clinical algorithms are race correcting for, things like kidney disease, that have significant impacts on how people receive care day to day. And we have understood, I think, in the last couple of decades, I would say, the ways in which, when used incorrectly, those types of race correction in clinical algorithms or race-based medicine can actually be deleterious to communities of color. We also can see that when representation is not talked about with nuance, there can be really negative impacts that are broader than just science. We saw from the Buffalo shooter in 2022 that a lot of their beliefs were not just grounded in cult-like beliefs or in dogma, but were using scientific papers to justify their beliefs and justify beliefs that support a white supremacist ideology. And so it's really important that in a space like precision health, but also genomic medicine, but also even just understanding genetics, that those of us in this space are ensuring that we're having thoughtful, nuanced, and accurate conversations about what genetic ancestry means versus what social constructions of race mean, because we know that when it gets into the wrong hands, and we have proof of that here, that it can negatively impact health, but also literally have social and cultural impacts, as we've seen with several racialized attacks on many minority groups in the United States. And so it's really important that, to underscore that, like, we do not believe that race is biological, and that sometimes when we're thinking about genetic ancestry without the nuance that's necessary, it almost inherently racializes biology without fully extrapolating what that means.
The final problem that we think about, and this problem is another issue of nuance, and also maybe even more than nuance, but something that just often gets completely left out of our scientific inquiries in genomics, which is that the genome and genomics may be the branch of science that's most closely linked to our human identities. So we often think about genetics as a blueprint for our biology. We often study the genome as a source of genetic variation that causes disease. But as scientists, I think we rarely, if ever, think about the genome as a source of richness and history that connects people to their ancestors, Um, uh, of late, I've heard some of my colleagues in genetics refer to things like Ancestry.com and 23andMe as recreational genetic testing, not to uh, to really belittle it, but just to see it as something separate from the type of science that we're doing in the field of genomics. And so it's clear that the role that genetics plays in someone's personal identity, their family identity, their family history is not something that we think about very much. And I don't think we'll get into it a ton in the data we show today, but something we find consistently in the conversations we've had with our research participants is that uh, they see DNA as being closely linked to their identity as people. And so this is a problem that we think is very important. It has real implications, as the other problems we've just identified also have. One is that when we're thinking about privacy, and the de-identification of research cohorts for which genomics data have been collected, we know that this de-identification process is not perfect or impenetrable. There's been multiple studies that have shown that research participants and patients from cohorts of de-identified genomics data can actually be re-identified using surname inference tools and publicly available data. So, for example, if your third cousin submitted a sample to Ancestry.com and that data is publicly available, it's not very difficult to re-identify an individual from a research study using those data. It does take some level of sophistication and specific algorithms, but the point being that your identity is at risk every time you think about sharing your DNA with research, and we have to contend with this as scientists and hopefully good stewards of people's data and identities. We've also seen something really interesting and uh, you know curious, to say the least, that is, I think, surprising to many scientists with the new sickle cell cure, which, for those who don't know, is a gene editing based cure for sickle cell disease, whereby folks who have sickle cell undergo bone marrow transplants, and they're given back um, bone marrow cells that have been genetically modified to correct a variant that causes the disease. And there was some really great journalism and reporting, and even some audio journalism that was published in the last couple of years that have talked to people who've lived their entire lives with sickle cell and are struggling with the idea that they might be cured, because sickle cell has become such a part of their identity, and such a part of their lives. And so um, it's not it's not always obvious that that pushing forward with something like a genetic cure for a disease is in the best interests of all patients that are living with that disease. And so um, very critical to integrate the lived experience of those who see genetics being so closely connected to their identity. And just to say, like, on the left here, this is a clip from um, the from Kendrick Lamar's music video for the song DNA. Um, this is a favorite reference point for me, in terms of how DNA and the idea of something being inside your DNA has made its way into our popular culture. But you don't have to look hard to find these types of references. My guess is that in the next week, everyone sitting here listening tonight will probably hear somewhere someone say, something about, you know, it being in your DNA to do this or to think that. And so the idea that this is really important for who we are as people is clear. And so because of really that last problem, we've seen a solution to all of these problems as a whole, as one that's really guided by people's lived experience. So we put people, we prioritize people, as our title suggests, and we've decided to develop a solution to genomics, to diversifying genomics that integrates the lived experience of people who are participating in the projects. That those projects are co-created to reflect the complex identities that people hold, um, and that not only are our genomics efforts focused on long-term health outcomes of people that participate, but also on the day-to-day well-being of those folks. And so one thing that we've done with our project from the very beginning, just in the way we work with each other and the way we work with our team, which is then extended to the way we work with research participants, is that we've established what we would consider an ethic of care, um, where, you know, your well-being is more important than us pressing forward with this project or collecting these data at the expense of your well-being. And so that's a value that we hold near and dear. These pictures, these photographs that surround those three key features of our solution are actually pictures of people that have participated in our work. And we actually put significant resource into photography from the beginning of this project, so that we could capture the humanity of those who've contributed so much to our work, and really to start to build new narratives around research participation, who's participating, why are they here, what do they care about, and putting a face to those data is extremely important for us, but is also going to just carry a lot of weight and hold a lot of power as we share these data into the world.
Thank you for that, Brett. That was really well said. So with that, this has been our approach. And so we call our approach, especially coming from my world as an ethnographer, and then both Brett and I's worlds as community engaged researchers, we call our approach CBPR meets ethnography. So thinking about community-based participatory research and it coming together with ethnographic method. And with that, we also think about how genomics education is important in how we approach the work that we do. And so to do this work, we launched in June of 2022, specifically on Juneteenth. And as we mentioned earlier, we had a slow start, right? We met in 2020, and we actually had anticipated starting this project in 2021, really on the ground, but there kept being waves of COVID that we wanted to be mindful of. And we also knew that we wanted to gather people in person, and that we wanted to be mindful of the fact that people's well-being is never on the line because of our research desires. And so we took our time until June 2022 to fully roll this out once we knew that it was safer for people to gather and for us to gather with others. And so to do this work, we consider this, even in our approach, we consider this a mixed methods project that had kind of 5 different active components. And so from the beginning, we engaged a community advisory board to provide us guidance and understanding around what do people want? How do we engage with communities in the St. Louis area? What are their concerns? Getting their feedback on interview questions, focus group questions, and also survey questions was really important to us before actually starting to disseminate all of these things into the community. And so then from there, we started really intentional in-person recruitment, hand-to-hand recruitment. Oftentimes as researchers, we know that like it is sometimes easier to use online resources and people you do not know to complete surveys. But for us, we really wanted to, one, demonstrate the breadth of knowledge that was in the Black St. Louis community, but also go to people where they were to make sure that we were touching a representative sample of the St. Louis area. In addition to our survey responses, we also wanted to make sure we were balancing our quantitative data also with qualitative data. How do we get deeper? We might understand like, okay, well, this is where people go to the hospital, and this is where how people decide about genomic sequencing. But what about the why? What are the root causes of that? What concerns people about this next wave of precision medicine, the questions that we couldn't necessarily ask in a survey. And so the focus groups really grounded us and gave us an opportunity to go deeper with individuals. And so we had 4 groups of focus groups that went through either 3 or 2 different sessions together to ask deeper questions first about ideas generally about genetics and genomics. Then we went deeper into health and precision medicine. How are people thinking about precision medicine and sickle cell? As an example, we had case studies, we had scenarios. And then the last focus group really focused on how are people thinking about their data and privacy methods and how that impacts their decision making. From that group of wonderful individuals, we then were able to do 40 more interviews with individuals to really even go even deeper to understand what people individually were having as health challenges, how they saw precision medicine being a useful or not useful or a benign tool for them in the future, but also what their everyday relationships with biomedicine and access to healthcare. And as Brett mentioned on the previous slide, it was really, really, really important for us to then capture these experiences, not only kind of capture the data collection process, but also make sure that we were capturing the real people that were a part of this, so that if we ever, and at the time we were wondering if this was going to be an if we were ever to actually sequence anyone's genome, we actually had a real living face to match with that, to show kind of the depth of lived experience and the humanity of the individuals that were deciding to be a part of the study. And one quick note that I'll just also make about the ethnographic approach. One of the things that was really critical in the ethnographic approach was that we were going to do long-term engagement. So that's really one of the key hallmarks of doing any type of ethnographic project, is that you don't just drop in and do a quick survey for 4 weeks and then run back to your office and then just, write a bunch of papers and say, oh, I have this representative understanding after these 4 weeks of surveys. But no, instead it's like, no, we're going to do a survey for a long period of time. We're going to do interviews and iteratively over a long period of time. And so from June 19th, 2022, all the way to December 31st of 2023, we spent a full 18 months doing this work, which again is, in some ways a very different than the typical research enterprise, because it wants us to work fast and, produce new data really quickly and then write the papers and get it out there, which is something that we do believe in for certain projects. But when we're thinking about rebuilding trust and repairing relationships and really making sure that also individuals who have been harmed by the very institutions that we work for, we need to also understand that we needed to prove ourselves to be worthy researchers and that people wanted to collaborate with, because that we knew that would impact their experience, which is why that ethic of care became so critical to our approach. So we would love, and so in the first phase of this work, we're going to just share like 3 highlights with you all today of some of the things that we found, and then also share with you the kind of phases that have come out of this rich data collection process. So one of the highlights that we highlight in a forthcoming paper that we've written is that trust in science and medicine is complex. And so what we found in our sample was that trust in individuals' primary care doctors was extremely high. However, as soon as we took that same sample of people, then asked them, okay, well, if that's your trust in primary care doctors, what's your trust in governments and healthcare institutions? And you can see a significant shift in our answers. We had over 80% of individuals say, yes, I trust my primary care doctor. But as soon as we started asking people, do you trust your government? Do you trust the healthcare institution that houses your doctor? Responses significantly changed. Now we only have 30% of individuals that still trust their government and trust their institutions, but we also now have about a little over 40% of individuals that are saying, absolutely not. I do not trust governments or the healthcare institution that may house my primary care doctor. And then a significant chunk of people who just simply were not sure. And so this tells us something really, really meaningful about how people are understanding their trust in science and medicine, but who the trustworthy actor is. And so from a focus group that one of the first focus groups actually that we had a participant share, quote, yeah, there's kind of this idea like you can't put Pandora back in the box. Like once that has been taken from you, it's not something tangible that you can take back. So really there's a whole lot of trust in giving that to someone. And in this moment of the focus group, the individual was really grappling with, if I do want to be a part of a research study and provide my genetic material, there are usually all of these different disclosures and things that I have to say of like, you can use my DNA in perpetuity. And as Brett shared earlier, there are so many different ways that people can be identified. So when we think back to the problem 2 and 3 that we identify, this issue is even heightened even more in communities that have experienced harm and gaslighting by the biomedical complex. And so it's really important that we're listening to the fact that people are not trusting their institution, their government and their healthcare institutions, but they are trusting their individual primary care doctors.
The second highlight here is that we found something that we were expecting to find, but that I think a lot of my colleagues in genetics and other colleagues throughout biomedical research may not have expected, which is that Black folks in St. Louis, they already value what genomics has to offer. So there's a tendency to assume or sort of presuppose that folks who are disconnected from healthcare and science, um, that they don't know about it, they don't know what they need to know about it, and that they may not even be curious to find out. And so I think our data show that it's actually quite the opposite. So more than 80% of the people that we surveyed stated that they were curious to learn more about their own genetics, and more than 60% of the people shared that they were interested in at some point getting their genome sequenced. Um, and so this just shows that, while folks are still not crystal clear on the process and exactly how to become integrated into the research world, that there is an appetite for it, that they are curious about it, and that they are interested in the use of genetics and genomics in research and also within healthcare. And so one of our research participants who had actually quite a bit of um, reservation about healthcare in general and shared over the course of multiple interviews some very negative experiences overall with the healthcare system, shared this quote with us, which was quite hopeful. And she referenced another focus group participant who had undergone some exome sequencing for her cancer treatment, and that sequencing those results actually changed the course of her treatment. And this is something she shared in the focus group. And this participant shares with us that, I feel like Miss X would be my saving grace. Like she would be the standard to where I would be like, this is why we need to do this thing, She gives me hope and makes me want to engage and trust in this thing, right? This is what's going on with genetics. I think the difficult part is that the people that could probably be supported by it the most don't understand what it is. So even here, among a person who's quite hopeful, for the future of this. Still some hesitation that that the folks who actually need it most may not have access to it. And so for us, this is the type of response or the type of lived experience that really drives us forward. The third highlight here is that not all precision health activities are acceptable. So while people are excited about genetics, clearly see the power of genomic data in health and medicine, there are boundaries that um, that they that they hold. So I showed on the last slide that more than 60% of people were interested in having their genome sequenced. If you ask folks about their interest in getting their genome sequenced for the purposes of their health, that number increases to about 80%. And then of those people who are interested in getting their genome sequenced, only about 40% are comfortable with new technologies to do genome editing. And so most people who are interested in participation in research and potentially even having their own genome sequenced draw some line around this intervention to actually go ahead and edit DNA as a potential therapeutic. And so I think this is really important to recognize that while folks are excited, there are still hesitations about the safety of their data, and then also in this case, the ability to intervene on one's genome. And so here there's a quote here from another focus group participant, just kind of expressing their willingness to participate, but then also the pause that they would have and the hesitation that they might also experience. So this person says, I think I'd be willing to share DNA in this case as a family member, but I think I'd be like, I'm going with a family member because I will be cautious about it. Just because I would want to know like what's going to happen with the information after and what are they going to do with it. Lots of conversation in our focus groups and interviews around um, what happens to a DNA sample once it's taken and sequenced? Does that DNA sample itself go into a freezer for eternity? Is it just the sequence that lives on a hard drive or in the cloud somewhere? Folks even expressed like concerns about giving blood and wondering like when they give a random blood sample, are clinicians actually using that to extract DNA and do things, behind the scenes that they didn't consent to? And so there's a lot of hesitation here around providing specimens and samples for science. Um, so
Awesome.
yeah,
Okay.
given the complexity of all these data and we don't have nearly enough time to share all of the qualitative findings that we have from our focus groups and interviews, but we felt it was really necessary to digest these data in community with many of the people that actually participated in our project, and then also think about how do we start to share these data and new ideas about genetics and genomics with more people in the community. And so Chelsea, you can share here about our CEC efforts.
Yeah, absolutely. So, what we call kind of, phase two of our project that we're kind of still in, but also kind of shifting into phase three is our community education collective. And so, what was really important and also is a value of a community engaged research approach is again, not taking the data and going to analyze it back in our office with just our teams, but also making sure we're doing it with the individuals actually impacted. And so, we developed two outputs of our community education collective. We had active genomics workshops, which is the picture on the left where Brett and I are actually outside at a park this past summer, teaching a genomics workshop on some literally responding to some of the concerns we heard in the survey and starting to provide education to individuals in the St. Louis community. And then we also created a data analysis team, which are the two pictures on the far right side, where we we spent several weeks actually teaching quantitative methods in R, but also qualitative methods of theming and coding to our community education collective. Some individuals were not a part of the project before. Some people were a part of the project. And so we wanted to not only gear them up with the tools to be able to analyze the data with us, but also to be able to participate in how we disseminated it and got it into the community. And so these are just a couple more pictures of how we were thinking about education and outreach in this phase. We've partnered quite a bit with an organization called Black Girls Do STEM in the past. We've also been out in the park and have worked, we quite literally table at events. Sometimes that tabling when we were in data collection was to recruit new individuals, but we've even continued tabling after that because we believe that it's important to continue to show up in community even when you're not making an ask. So making sure that we're then disseminating genomics brochures and information that we've developed. Also, we're about to have in a week our 3rd annual Family Appreciation Day, but the picture you see on the top right is a yoga class that we had with an organization called Wellwood Woods that came in and actually taught, wellness and breath work activity with members that have been a part of the BGP family. And this last phase, phase 3, is our public dissemination phase that we're also kind of gearing up into, that I'll let Brett share a bit of before we wrap up, because I'm looking at the clock and I want to make sure we're leaving time for some questions.
Yeah, so this is something that came out of our community education collective. Was the idea that we wanted to share the project itself, but also the data from the project and the findings of the project and what they represent with a broader collection of people from Black communities in the St. Louis region. And so the idea of curating a museum exhibition came up and is becoming a reality. We have a grant through the Institute for Clinical and Translational Sciences at WashU, supporting the co-curation of a museum exhibition with a team of community members, many of whom actually participated in our data analysis sessions. And the idea here is that, rather than just squirreling our data away in scientific journals, which we, as Chelsea said, will do, it's really important to share these data in new ways. And we've decided that using art and multimodal forms of art as a vehicle for sharing data is our path forward. This was really inspired by some of the graphics that sociologist W.E.B. Du Bois produced with his students for the 1900 World Expo. An example of which is shown on the left, where taking basically census tract data or census type data and producing these very intricate hand-drawn pie charts and graphics that don't resemble our traditional pie charts and graphics, but add a bit of a twist. And so we were inspired by this, and we're continuing to work with a team on a monthly basis to curate this exhibition. On the right are a couple pictures of our junior curators who are creating vision boards to help us think about how this exhibition is going to come together. We continue to brainstorm with our research community about what's next. And we're extremely committed to following the desires of those who've already participated and those who are not part of the Black Genome Project, but are part of other research projects that we've been working on. And so Chelsea mentioned early on that she's been working for many years in the world of ALS. And so we've developed some ideas around how do we integrate mixed methods, qualitative methods, and sort of equity-focused initiatives into more traditional disease genomics projects. And so in the last minute or two here, Chelsea, you can share a little bit of our thinking on how we move into maybe sequencing a cohort of Black ALS patients.
Absolutely. Thank you, Brett. So the rare disease genomic space is where we're thinking about next because of the work, as Brett mentioned, that I've previously done. So my lab at Yale, the Leaf Lab, is a unique space where we focus specifically on health disparities and health inequities in rare disease and in specifically rare neurodegenerative disease. And so right now, a lot of that has been looking at ALS. And so we've worked with Morgan State through one of our lab's community advisory board members to amplify how ALS looks in the Black community. We've also developed the Possibilities Grant Program, which provides up to $10,000 for individuals living with ALS or their caregivers. And through this program alone, individuals like Miss Asia Jamie have been not only awarded funding, but also are helping in a community engaged way with the epidemiology of understanding ALS. And so the individuals, ALS has a perception as being a disease that does not impact as frequently people of color. But we're realizing through this program that we went from, 8 months ago only having 3 applications to now being having an influx of over 100 applications. And so we think a lot about the opportunities that are then created when you're also thinking about some of the genetics in ALS that could be studied further to better understand disease-causing genes in particular conditions. And so this is just a quick snapshot of a family that has been impacted by familial ALS. All the individuals in this picture have or had the SOD1 mutation. But one of the things that's really interesting about it is that even in the younger generations of individuals in this family, despite having a very clear genetic cause of ALS, which is unique because 90% of ALS is sporadic, there still was misdiagnosis. They still were not being diagnosed timely. And so it shows that even when you have your own family history of a genetic disorder, given the barriers and injustices that can occur in our biomedical system, but also in precision health, it can impact how fast someone is able to get care and then be able to get treatment. And so individuals with the SOD1 mutation, there is a medication specifically for those individuals called tofersen that many individuals in this picture were very delayed in being able to get in the last 3 to 4 years because their diagnosis did not come on time. And so I will just end by saying that, we do not have all the answers. And even though we've identified some of the problems and also we believe we have some of the solutions, we believe more than anything that this work has to be done in concert with community members, particularly I think coming back to the fact that our genetics and genomic medicine are some of the most closely linked ideas to our identity. And so being able to work directly with community members not only helps us improve diversification in larger databases, but also helps us make sure that we are using nuanced approaches to really making sure that we are making sure that precision medicine isn't just available for a set of lucky people, but is actually available in the future for all. So many thank you to our funders and our institutions, WashU and Yale, that have supported this project a lot. We shared 2 pieces on this last slide that we encourage you to read about how we're engaging with this work. We wrote a short op-ed in the Lancet Regional Health about our diverse databases actually creating equity in genomics. And then we also wrote a shorter piece in the St. Louis American, which is specifically a Black newspaper in the St. Louis area, around the future of precision medicine and our work in public health genomics, and taking a group of high school students down to Atlanta to actually co-present with us on our research. So thank you so much to everyone for being here today and listening to our work, and we'd be happy to answer a few burning questions before we wrap up.
Thank you both so very, very much. I will try to get to questions. There's two that I'd really like to see if we have time, but otherwise I'll see what we have time for. And I did want to make a very, very fast shout out. So thank you so much for that wonderful presentation, and to mention the podcast where I first learned about you. It was the Genetic
right.
Frontiers podcast.
Yes.
And I heard about your ethic of care and prioritizing people, and I just was so impressed. And we thank you so much for your time. So I will quickly get to questions, but if folks can check out that Genetic Frontiers podcast, it's totally dedicated to genetic science. And I think that you'll find a lot of good discussion there.
Absolutely.
This was so much to unpack for those of us who really believe in precision medicine, but also want to make sure that we're adhering to protections, especially for vulnerable populations, but for the whole population in general. You mentioned so many things that were concerning to me, things like the flawed de-identification system, or what happens if the company folds and you gave them your DNA, or where does things get stored and for how long, and all these questions. So one of the questions asked was, they even asked for DNA at the dentist office. How safe is providing DNA within a clinical setting? So are there distinctions between these kind of, companies that are doing it more commercially versus other settings?
Yeah, this is a great question. Really important distinction there. Yes, they are different. Of course, you know, in a commercial setting, the one of the biggest risks that to be aware of or considering is sort of what happens to the data. Are the data an asset that company can basically sell to third party to third parties? And in many cases, the answer is yes, that the database is an asset that a company can actually share or sell with others. So they're supposed to be doing that in a de-identified way, but as I pointed out, like, none of this stuff is ever really truly de-identified, so that's a risk. In a clinical setting, the DNA is being shared for usually a very specific purpose or a very specific question, though there is a trend towards like broad IRBs that essentially create biorepositories that are sort of open source, that researchers at that institution or even at other institutions have access to. And so as those kind of open IRBs or open consent platforms become more popular, I think that being vigilant in the clinical setting is as important. This is something we've discussed with our research participants quite a bit, and we've sort of run different scenarios to help kind of parameterize, like, what is it that people truly feel comfortable with? What are the features of a consent process that folks really care the most about? And for example, like, every time someone accesses your genome sequence, do you want to get a little ping on your phone that you have to like consent to, or would you rather say like, no, I don't want that bother? You can have it, and you can use it for research purposes. So
Thank you.
definitely differences between commercial and clinical, but as I mentioned, clinical and research settings are becoming more kind of open source in their structure.
Yeah, thank you for cautioning the vigilance. And I don't know if you're going to have time for this final question, but I'm going to throw it out there.
Yeah, let's
You
do it.
mentioned,
Yeah.
And just on solutions now, you mentioned a lot of things like, I'm curious from a recruitment perspective, from research recruitment, and with rare diseases like CADASIL, and it's difficult to get wide representation. We try to capture real people. We try to, some of the things that you discussed really resonated with me in terms of making things more inclusive, how we maybe disseminate the data more visually, or try and do, all of that. But do you have any suggestions in terms of recruitment? Because it sounds like the trust issue, it's not about their not being interested in participating or learning or volunteering, but what can we do to quickly maybe touch on other ways that we can repair trust when it's hard enough to reach a rare population to begin with, let alone a vulnerable one.
Absolutely. I think that, I think one of the places to begin is ensuring that when we're talking about community recruitment, we see ourselves as a part of that community. I think a lot of times we often see researchers kind of fly into a community, try to recruit a bunch of people, and then fly out. I think that this is why long-term engagement is really important to us. And I think that that can happen in person in community, but it also can happen in virtual spaces. I think that like really being interested, not because there's now a new study, but like saying there's maybe a study happening in 18 months, but like we know we need to start seeding the steps of that now. So are we going to like virtual support groups? In communities that we're interested in? Are we finding rare disease organizations that particularly have a equity and a justice focus that we can support? Are we also supporting those organizations more than just in more with more than just our presence? Like, is it also a symbiotic relationship where it's not
Yes.
just here's what we can get, from the research, but also what are we providing? And beyond
Yeah.
just like I think sometimes we think about as researchers, like, oh, we just have to provide an incentive and that will get people to come. No, that's absolutely not true. Like sometimes the incentive is not even enough, right? So it's like, are you supporting that organization? Are you co-working with that organization to maybe do a webinar and show that there's a real meaningful interest? So I think that like making sure that as a researcher and even as a recruiter that you are engaging in communities that are hard to reach because there's a depth of care and not just because there's a transaction to be had.
Yes.
I think is really powerful and I think is an important first step. And I would really encourage, I think specifically in the rare disease community, because I absolutely empathize with like, and that was like kind of one of my challenges when I first started my doctoral program, was like, where do you find the people? Where are they at? But the longer I was in community and had conversations, it really helped bring me to the right people. And so I think that's where I would say is a great place to start and build those relationships long term.
Yeah, thank you so much. I really hear what you're saying in terms of earning trust.
Yeah.
Right. So that is like really where it's at. It's like that's how you repair it, is that you earn it. And so being part of the community, and I'm definitely proud of this CADASIL community in terms of the interactions between the researchers and the community and the advocacy groups and the fundraisers and the way that they are making an effort to work together, not in a transactional necessarily way, because the financial incentive is definitely meant to be non-persuasive, and these folks are not motivated by the financial incentive. They are motivated by the science, by helping family members, by doing what's right for moving things forward. So thank you for this beautiful reinforcement of where we go from here and what we can all be doing together. We thank you so much. It was a wonderful presentation.
Absolutely. Thank you. Thank you all for being here and for that feedback.